Okay everyone, hello, and I apologise initially if the sound quality isn't quite as good as you're used to on Mr. Gill's science stuff, but that's because I'm not in my usual recording facilities, so I've cobbled something together elsewhere so we can get some of these videos done. Okay, so this one is aimed at Year 12, A-level, cell cycle and mitosis. So basically we're going to look at this video, how and when a cell divides, okay, and this is the first of a couple of lessons on this topic. So, first off, this is what we call the cell cycle. Now, for the vast majority of a cell's life, it is not, I repeat, not in mitosis, okay? The vast majority of the time, it is just sitting around doing its job, okay? Mitosis is quite rare, okay, in a cell's life, particularly in an organism so complicated as yourself. So, that little red bit at the top, that M, that red bit, that's the bit we're going to come to next. That's the actual act of cells doing division, right? This so-called G1 gap and G0 gap here, these are the bits where the cells are just increasing in size, they're producing RNA and proteins, they're doing their job, basically. Okay, and obviously there are various checkpoints in here to ensure that the cell is doing that correctly, and if it isn't, then it isn't going off around mitosis again. G0, of course, there, look, that's the time when the cell will leave the cell cycle and absolutely stop dividing. Okay, the cell will just do its normal job. Right, and again, there are checkpoints to move through these stages. So if a cell is signalled by an external stimuli to divide, okay, then it moves through that G1 into what we call the S phase. Now, S phase is called the synthesis phase, and that, of course, is where DNA replication is taking place, because the first thing that needs to happen if a cell is going to divide is you're going to need a full extra set of the genetic material in there to allow it to do so. Okay, so from then, that takes as long as it takes in terms of how much DNA the cell has got. If it's got more DNA, it's going to take longer. Less DNA, it's going to be shorter. And then you get into this other stage, this one up here, called the G2 gap, which basically is where the cell gets really ready to do its dividing. So essentially it's growing, it's producing new organelles, producing specific proteins required for mitosis, and essentially that last final checking process to make sure that everything is okay. Now, a word you might have come across at GCSE is this one down here, which is interphase. Now, interphase basically is the combination of the S phase and the G1s and G2s. Everything that isn't mitosis is referred to as interphase, and you do have to know that word. Once we've gone all the way around that cycle, then we get into mitosis, which is the bit that you're most likely to be asked questions about. But as I say, for a vast majority of the time, the cell is not doing mitosis. So, I should clear up a really important point that some people get wrong at GCSE, and it doesn't matter so much at GCSE, but it really causes a problem at A level, right? Massive issue, and that is, what is a chromosome? Okay, now this structure over here, right, that we see is what we probably always think of as a chromosome, right? That X-shaped thing, and it's true, it is a chromosome, but it's a chromosome at a very weird point in its life. Okay, and that weird point in its life is during cell replication. For the vast majority of a cell's life, it does not. The chromosomes do not look like this. Okay, because as we've just seen, what has to happen before mitosis is the DNA doubles up. So this is actually twice as much DNA as you would always, or as you would see normally. Okay, this is a chromosome most of the time. Just one of those sides, okay? Now, this is confusing because at GCSE we teach you about the word chromatid, which only applies when the cell is like this, when the chromosome is like this, okay? We also get another confusing phrase, which is a pair of chromosomes. Now, this over here is not a pair of chromosomes, right? Now, let's get that absolutely clear. If you thought that at GCSE, you probably would have got away with it, and that's fine. But it is not, repeat, not a pair of chromosomes. That is one chromosome. This is also one chromosome. The only difference is that this chromosome has been through the photocopier to make this chromosome. So there's two copies of identical DNA. When we say a pair of chromosomes, what we mean is the chromosome 1 that you got from your mum and the chromosome 1 that you got from your dad, which will have the same genes but not necessarily the same alleles. And they never ever look like this, okay? they never sort of look like a cross shape. That is one chromosome. So can we be really, really specific? At the beginning of interphase, this is what our chromosomes look like, just a single linear piece of DNA, okay, usually wrapped around a lot of proteins called histones. And then over here, look, we've got our original DNA, we've got our copied DNA, and they're connected together at a thing called the centromere. Okay, one chromosome. This is one chromosome, that's one chromosome. It's just been through the photocopier, not a pair of chromosomes. If you've got that in your head, break yourself of that habit immediately, or the rest of this house of cards is going to come crashing down. Right, moving on. So time. As I said before, a lot of this depends purely on the amount of DNA that the cell has. So I'll give you a couple of examples here. Bacterial cells every 30 minutes, skin cells about every 12 hours, right? And that's about the fastest dividing cells in you. Now the reason for this is bacterial cells do a slightly different process called binary fission that we'll get to another time, and skin cells obviously have got way more DNA. Okay, now lots of cells don't divide very often at all. Liver cells on average, this is a rough average, about every 2 years, and nerve cells may, once they finish dividing, once they're there, never divide again. Hence, if you lose your brain cells, they don't come back. Okay, but obviously there's a lot of differences within the nervous system as to how that behaves. Okay, so it's variable, and it pretty much depends on what the cell's job is and how much DNA the cell has got. But your very fastest cells going through cell division would be bacterial cells, 20 to 30 minutes. The fastest of the human cells would be skin cells, because you're constantly making and losing those. Right, so here's a summary of the phases you need to know. Now at GCSE, all you will have learned probably is that the DNA doubles up, and then the chromosomes line up, cells split up, and the cytoplasm split, and that's fine. Okay, absolutely a-okay for GCSE. Now if you had a GCSE teacher like me, they might have gone somewhere a little bit further than that, saying there's actually a bit more going on, and actually it looks a bit more like this lovely background picture that I've got here that was taken by one of my students a couple of years ago down the microscope. Okay, that there's actually a bit more complexity going on, that there are some cells that sort of the chromosomes become visible here, then there's like a lining up phase over here. Most of the cells, by the way, are not doing anything of any particular interest. If you are an A-level student, you will try and get these pictures yourself in a required practical. But the summary goes as follows. The phases all have names. We've already discussed interphase. Interphase is everything that isn't technically mitosis. Okay, so interphase is growth and DNA replication and checking, making sure the cell is ready to go through mitosis. Then you have prophase, where the chromosomes become visible, metaphase, where the chromosomes line up, anaphase, where the chromatids making up those chromosomes split up. At this point, the naming gets upgraded, right? You can start saying what was a chromatid is now a chromosome in its own right. And then telophase, where two separate nuclei form around the piles of new chromosomes. And then cytokinesis is where the cytoplasms split up. And you are expected to know those words in that order and what happens in them. But I have a handy acronym for this. Just remember IPMATC. Right? If you can't remember that, what's wrong with you? It's the most handy, convenient, memorable acronym for there. IPMATC. Right? Some people like to make up other ones. So, for example, if you ignore interphase and just look at the prophase, metaphase, anaphase, telophase, you get PMAT. And my head of department likes to do a graphic mime of peeing on a mat to help her students remember. Okay, if it works for you, it works for you. Great. Or please make up another sort of acronym of your own choice. Okay. So let's go through the phases then, right? So not only are you expected to know what the names of the phases are and roughly what happens, you're supposed to recognise them from microscope images or from diagrams and be able to identify them, put them in order, say what's happening, name the bits inside them. Okay, so as we say, in interphase, the cell's not dividing, you've got normal cellular functions. The chromatin, which is that combination of protein and DNA that makes up chromosomes, is not visible. It's all unraveled, unreeled, so that the DNA can actually be used. The DNA, the histones and the centrioles are obviously all replicated. Now, of those things, you will have heard of DNA. Histones, as I said, are the proteins that DNA is wrapped around, and the centrioles are these little sort of these little organelles that are going to produce some spindle fibres. These little fibres that help pull the chromosomes, chromatids around the place. Okay, and you get replication of all the other organelles. So you're going to need some new mitochondria, you're going to need some new ribosomes, and so on and so forth. Right. Once that's happened, and that technically isn't in the series of mitosis, you get into prophase. Okay, now prophase is when you can first see the chromosomes. Okay, so the chromosomes condense and become visible. Now bear in mind they've already been through DNA replication, hence we've got this double chromosome appearance, right? But that is not a pair of chromosomes, right? Just to reiterate, you can see the labels there on the diagram. Okay, so chromosomes condense, become visible. These centrioles literally split from one point on the nucleus to the opposite poles of the nucleus, and the nuclear membrane disappears. Nuclear membrane breaks down. So at the beginning of prophase, you'll have a nuclear membrane there. At the end of prophase, you won't. So sometimes you see them written as early prophase, late prophase, or prometaphase, or intermediate ones. For you, as A-level students, prophase is just fine. Okay, so there's the photo down the microscope. You can see that's quite a late prophase because there's no nucleus in that picture. Okay, but you can see the chromosomes, right? They're not particularly organised yet, right? And there's no nucleus holding them together. Okay, next, metaphase. This is where the spindle fibres, these little microtubules that come from the centrioles, send out the little spindles and connect to the centromere of the chromosome, and the chromosomes line up along the equator. If you can find a good metaphase down the microscope, you've done really, really well, because this is a very quick process. Once they're lined up, they move on to the next phase very, very quickly. Okay, so lined up along the equator is the telltale sign of metaphase, and spindle fibres have formed and pulled them into the right position. Okay, you can see that there. That's a perfect example of a metaphase, a lovely, lovely image of a plant cell in metaphase down the microscope. Okay, so that's metaphase. Moving on, anaphase. This is where the centromere is split. So the chromatids separate, right, and they're pulled apart. And I always like to think of this like, I don't know if you've ever done that fairground game bungee running, where you have a harness on and a big elastic thing on your back, and you run down a bouncy castle and try and put a Velcro thing as far away as possible. And when you get pulled backwards, eventually by the bungee cord, you end up in this weird V shape of like that, with your legs and feet, legs and hands sticking out in a big V shape, being pulled back by your waist. Okay, this is pretty much the shape that we've got here. So the fact that the chromosomes are in that little V shape, and they are called chromosomes now, because once they've been split, right, they get upgraded to be chromosomes in their own right, no longer just chromatids. Right, so they get pulled apart, and that little V shape you can see there as they move towards the poles is very indicative of anaphase. So in anaphase, you can see them in V shapes being pulled towards opposite poles. Now this is done by the centrioles, and mitochondria, of course, provide energy because it's an energy-hungry process. Right? So if we look down the microscope there, look, that is a lovely, lovely example of that stage. Okay, you can see them in the V shapes being pulled apart. Right, that's a lovely anaphase there. Right, next one. Last one of the main sequence of mitosis is telophase. The spindle fibres disperse. We've got our, so they go away. You get your nuclear membranes forming around each set of chromosomes. The nuclei reform. Okay, and you start to not be able to see the chromosomes down the microscope because they start to unravel, getting ready to actually be used to do their job. Okay, so the visible chromosomes kind of fade, disappear, and very temporarily you've got a cell that sometimes has two nuclei in it. Okay, so that's a very late telophase and an early cytokinesis, which is the next stage. As you can see, the cell membranes are starting to pinch in and starting to split. Okay, so the last one, cytokinesis, is a little bit different if you're talking about animal cells versus plant cells, of course, because there's the complicating factor of a cell wall in plant cells. Okay, so in animal cells, you literally get this ring of filaments, this ring of the same sort of microfiber filaments that go round, right, the sort of middle bit round the equator, and they just pinch that off like splitting up two bits of dough. Right, so they tighten up, and split the membranes, and that's the cell split in half, no problem. In plant membranes, in plant cells, that couldn't happen because it's not just a membrane, it's a cell wall as well. So what you actually get is you get new cell wall laid down by these beautiful little vesicles that bring loads and loads and loads of cellulose into the middle, and they form this kind of chain of cellulose. They build a new cell wall inside the cell, which eventually splits the cell into two. Okay, so that there is a lovely late cytokinesis. Or, if you're feeling clever, because this is a cycle, we could also say that that is a interphase, of course, because it is a cell cycle. And the sort of definition between when cytokinesis is ended and interphase is started is a bit of a blurry one. Okay, when you look at these down a microscope, you do have to make a bit of a judgment call. Okay, so we could say that's a late cytokinesis or an early interphase. Okay, right, so those are all the stages. Okay, and as I say, just remember the handy acronym IPMATK. You've got to say that quietly because cytokinesis isn't technically part of mitosis. Right, maybe I should have a small I as well because neither is interphase. And the main stages there: prophase, metaphase, anaphase, telophase. IPMATK. Very straightforward. Right, so just as a conclusion, a summary here, look, what you should know from this, and you do need to go away and put this in your brain, unfortunately, you need to know the names of the phases, you need to know what order they occur in, where they sit in the cell cycle, how to recognise them from pictures or diagrams, and then how to put them in order, of course, and how to describe the stages using the correct cell terms like chromosome and chromatid in the right place at the right time. Okay, so if you can get all that, then you are well on your way to understanding mitosis. Thanks very much, and I'll see you again next time.